Thank you. Very interesting philosophical talk about function. And I think that if somebody generates this artificial general intelligence, then we're all doomed. And some computer scientists actually believe that soon general intelligence will be not necessarily based on large language models, but if not, we can still strive to find. So this is not about large language models. Also, we also working on like protein language models to predict some protein properties. But about genes of unknown function in fungi. Uh, so I am from Joint Genome Institute. Just a few words about this institute. It's based in Berkeley, part of Berkeley Lab, founded by Department of Energy. Sorry.
I can hear you.
Sorry.
Not too bad, but this is okay. This is a good distance.
Yes, so it started like sequencing facility, but now expand to these multi-omic studies of fungi, microbial communities, plants, to the project relevant to DOE goal of addressing environmental and energy issues. So one of the program is fungal program. This is page for its portal microcosm where fungal biologists, mycologists send proposal to sequence their favorite genome. And we have now like close to 3,000 fungal genomes annotated. So this is already mentioned by people previously that roughly about 40% genes of unknown functions. This is on PFAM, but it's roughly the same approximation maybe by other approaches. So different fungal clades have different percent of unknowns, but roughly it's 40%. And unknown genes are, of course, important. So, for example, in a recent study of Basidiomycetes clade of fungi, about 100 species, we just look for machine learning to discriminate between different lifestyles. So they mostly are either plant pathogens, so kill plants, or saprops, live on dead matter. And when you build some rather simple machine learning algorithm, it's quite good predict based on genomic sequence lifestyles. And when you're looking at features, most are just clusters of unknown function. So we just built these clusters of fungal families of unknown functions, just standard method MMC clustering, required two conditions, conserved across large phylogenetic distances, so present at least in 50% of fungal genomes or 90% of large fungal clades, and function is unknown, no PFAM, no meaningful annotation by some annotation methods, for example, eggnog. And automatic clustering, of course, it's always have some errors, so we could see, so removing outliers in clusters. And I should mention that most of these gene families are from single fungal species, but some have duplication, so it's not entirely orthological set, have some parallels. Looking on so in total we have 339 unknown gene families. Looking at taxonomic distribution, about half of them are fungal specific, happen only in fungi, not everywhere. Second largest class is genes which happen only in fungi and plants. And taxonomically, it's surprising because plants are kind of distant from fungi. Fungi is more similar to animals, metazoan, epistacons like we. But at the same time, it makes sense because fungi and plants depend on each other, and so they form symbiotic relationship. And some fungi pathogenic to plants, and some live inside plants, endophytic fungi. So it makes sense that, and they not largely explored. When we compare with other related databases, so there are some database called unknown genome, which is human based and based on clusters from Panther database. So they define this knowingness measure, which is assigned to each cluster as the largest number of Go terms to assign to at least one member of the cluster. And they counted 133 clusters with knowingness 0 and have at least one gene from humans. So when comparing with other fungal clusters, we just have 2 families. So as mentioned, it's not much large unknown clusters in animals and fungi. Additionally, some fungal clusters are linked to other clusters from this Panther database, but they have knowingness, it's very little. So it's Go terms like binding, catalytic activity, not real function is known. Another recent database, AFGB, which is based on clustering of these alpha-fold structures. So they define dark clusters which have no fold known. And when we compare fungal hypothetical clusters, in general, they correspond, each cluster corresponds to median of 8 AFGB. And why? Because AFGB, by some reason, are very small. So median number of cluster is 3. So I don't know, maybe have the definition of specific fold. And about 20% of the fungal clusters correspond to dark clusters, other like mix of dark light. Light, I mean, they probably have known fold, but function still not known. So these resources call for this our community of fungal mycologists to look experimentally on the function of these proteins. So we created some portal for these clusters of unknown genes. And so example, for each cluster, location on gene on some possible evidences, like for example, using HH bleeds to have distance homolog, and they presented in the portal, predicted alpha fold, and run again fold seek to have some possible homologs in PDB. Just 14 members have homologs with PTM more than 70. Some structures are very low quality, so we estimate that quota probably has disordered regions. And so, of course, we try use like recent computational methods to assign function, like yesterday or today presented. But we noticed this curious thing in 2 recent Go term prediction methods, deep fry and protein info. They make very different Go term predictions. So when you run its on all proteins from hypothetical proteins, so for example, deep fry, 82% of predictions are unique to deep fry. It means it's predicted on protein, but not predicted by protein info. And similarly, protein info also have 54% like unique one. But if you look even on proteins, when both predictions are made, just 3% of Go terms coincide. So kind of interesting, maybe. But of course, it's bulk prediction and many of low confidence. But if we take even like 50K high scoring predictions, or even 25K high scoring predictions, is still it's most of them unique and little overlap in Go terms. We're doing this to assign possible function to clusters if at least 50% of members of cluster have the same Go term. And not surprisingly, just for one cluster we have same assignment. So potentially function also may be inferred from these large-scale studies, like this, for example, joint collaboration between JGI and UC Berkeley, where it's called nutritional encoding for model fungi Neurospora crassa. So Neurospora crassa, like all phylogenetic filamentous fungi, it's very efficient at plant degradation, and many pathways for plant cell wall degradation many enzymes and transcription factor regulates these pathways are known. So we conducted this transcription profiling of neurospore cluster, and some 7 gene transcription factor gene knockouts on different carbon sources, from simple sugars, more complex sugars, or like the whole plant biomass. And at the same time, we have this DAP-seq technology for finding genome-wide finding of transcription factor binding sites. DAP-seq is kind of similar to ChIP-seq, but more scalable. It's a tool used at GGI for mapping transcription factor binding sites in many species. And 25 these encrusted genes are present in 25 hypothetical clusters, and 15 of them looks like regulated by 3 transcription factors. Evidence like their promoters have corresponding transcription factor binding site, and expression levels significantly decreases at gene knockout of corresponding transcription factor, and gene knockout specifically on specific carbon source. So, for example, 8 genes looks like regulated by this transcription factor KLR2, which is major regulator in the construction of cellulose. So it's significantly decreases in expression on when on cellulose. But this is, of course, not molecular function, but maybe process in participation in plant cell degradation. But anyway, some hint. So other study, joint collaboration between GGI and UC Berkeley, it's this TN-seq experiment on oleaginous yeast, Rhodosporidium turuloides. Oleaginous means it's produces a lot of fat, lipids. So, and TN-seq is a random insertion of transposon in the genome, and sometimes they disrupt, and frequently they disrupt the genes when they enter inside the gene. And this specific type of TN-seq called RB TN-seq, random barcoding. So every transposon have random barcode. When inserted in genome, we know its location, and by PCR sequencing, we estimate abundance of this strain, which have specific mutant for particular genes. So for 77 genes for this yeast, we have like estimate fitness phenotypes. But so Berkeley Labs have a fitness browser, which is linked to corresponding genome page of this yeast. For each affected genes, it's estimates under some condition how the abundances change, and it's like fitness score. So it's like low likelihood based on abundances in some particular experiment relative to wild type. But so we have about 10 genes from this genome in hypothetical clusters, but unfortunately they don't have significance in fitness score. So we cannot deduce. But other feature is co-fitness analysis. So correlation across multiple experiments with different genes. And it's estimated that significant correlation is if it's more than 75. But for 7 of genes we see, but it's still, this conserved hypothetical correlates with other hypothetical many. So hard to get function, but potentially it may, if we have more TN-seq experiments for different fungi, it may give some clues. So again, this is resource for fungal user community, and some people already using this resource. So for example, Inge Mortensen and Fabiana Contenses from Denmark, they're interested in antifungal drug development. So they took 10 genes from hypothetical clusters, mostly from Arabidopsis, because CRISPR-Cas9 tools available for Arabidopsis. Aspergillus. And they want to find genes which are essential. So if you knock out it, fungi dies, and so it has to be present in many fungal species and not in human, and our data kind of supports this. And so for example, Michela Amali, she investigates this early divergent fungi, which is quite different from bulk of fungi in many features, and she wants to find novel functions, for example, novel Cas enzyme, carbohydrate degradation enzymes. So maybe there are some enzymes exist which not present in current fungi. So this is ongoing work. So in summary, we have 3,039 conserved fungal gene families. We try to annotate it with all possible state-of-the-art computational methods, but of course it gives some hints, and proper experimental characterization is also needed. And this is a resource for GGR fungal community to focus on conserved unknowns. Thank you. So these two eagles also worked on this project.
Thank you.